So a couple of Punnett squares here for you. Lay them out kind of like I have. You'll see that there's like the square, but then you also want to be able to determine what are the ratios, the genotypes, and the phenotypes for each of these crosses. So if I have a parent who is homozygous dominant for the yellow eyes, like the yellow whites of the eyes, and homozygous recessive, meaning they have whites of the eyes, what are the ratios that I would expect of this cross? What is each genotype and phenotype that could result? Go ahead and do that same thing with the wings and no wings, which is I. Quick note, always make sure that it is very clear what your symbols represent. Note that I have my eyes clearly like eyes and not just lines. All right, so when we are looking at Punnett squares, and I want to make sure that this is really kind of making sense, we know the genotypes of the parents, or you have to be able to determine that at some point in the process. The whole purpose of a Punnett square is just to figure out the likelihood of their offspring having a certain trait or genotype, okay? So the only purpose for Punnett squares is kind of for predicting, almost like predicting the weather. But once you know what the weather is, it kind of loses its point to some value or to some degree. So as we're going to be doing today, we want to be able to figure out ratios, but then when we're done, we're also going to make a baby. So let's go ahead and start out with this guy. If we're looking at parents who are homozygous dominant and homozygous recessive, in other words, homozygous meaning two of the same allele, for the dominant one, big A, and for the recessive, little a, little a. One thing that I do want to make super duper clear, the biggest mistake I see when people are setting up their Punnett squares, please remember that this is an individual, right? Two alleles, meaning it's representing a diploid individual. That individual is going to have gametes. You know that 50% of the gametes will be from one allele, 50 will be from the other. This individual is big A, so half of their gametes will get the big A, half will get the other big A. When we put this together on the Punnett square, make sure you keep each parent on their own side. So do not do this, like, I see this a lot, where you end up with having the parent kind of get split apart. So one parent's big A, and so people will put it on both sides and then put the other parent. Don't do that. Don't. That's wrong. Very, 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 very wrong. So instead, what you always want to make sure to do is set up the two possible gametes that one parent could have on one side and the possible gametes that the parent could have on the other side. And so what we get from this is 4 possibilities. Possibility 1 is that the offspring that they have is big A, little a. Option 2, big A, little a. You see the pattern here, right? In this case, we're going to end up with 100% big A, little a. That is our heterozygote, and it will have the yellow whites of the eyes. Did we all get that one? Are we feeling okay about this so far? Okay. So as I go through on the next one, same general process. I clearly did not give myself nearly enough room. It is our farm. Okay. Big eye, little eye. Big eye, big eye. So the 2 genotypes that I could end up with are big eye, and big eye, little eye. That's going to make 50-50 or 1/2. But in both cases, my phenotype is going to be a dragon that has wings. All good so far? Let's do one more of these, and then I want to come back to Mendel's laws. Because Mendel had 3 laws, dominant and recessive. So this idea that one could be dominant and one could be latent, just kind of hiding and only popping up when there was no dominant one. So the law of dominance. Let's try big A, little a. What happens if I take this this pairing, okay, that's the parental one. They had all offspring who are heterozygous. What happens if there's some inbreeding going on and we get two heterozygotes mating with each other? What is the result of that going to be? And this is what is so remarkable about Mendel's studies, is that he connected what he saw the way that he did. That makes sense, right? That makes sense. So we have essentially two dragons. They have wings. Their parents had wings or didn't have, no, this isn't wings, eye color. Yellow, yellow, white. So the grandparents were either yellow-eyed or white-eyed. The parents are both yellow-eyed, but they're heterozygous. And so what Mendel realized with peas, what we can now see with our dragons, right, is that that recessive trait did not just disappear. It stayed its own separate entity. It could interact within an organism to make a phenotype, but it stayed separate. In other words, in this generation, what do I end up with? I get big A, big A, big A, little a, and I do end up again with some individuals who have that recessive trait. It doesn't disappear. It comes back, right? It stays there, but then becomes visible when in the absence of the dominant. What is this law that says we're not blending stuff together? The recessive ones aren't disappearing. It's just that all of the alleles stay their own separate entities. Do you remember what law that is? So this is Mendel's law of segregation. It said that each of the traits are their own unique entities. So if I have two heterozygotes, if I have two what he called hybrids, two individuals that have both versions of the gene, and they mate, then I'm going to see some of that recessive trait coming back out again. Okay. So I want to go over Mendel's last law. This one is not so fun. Actually, it's super fun. I really like it, but it's, yeah. Okay. So we're going to take a quick look at the law of independent assortment. When we were looking at meiosis and the chromosomes were lining up on the metaphase plate, it didn't matter which way one chromosome went. They all kind of went their own way. So just because you flip heads one time doesn't mean you'll flip it again, or doesn't mean you have to flip tails the second time. Okay. So what he said is basically each gene assorts independently of everything else. Getting one allele doesn't mean you're going to get a different allele. So for example, let's start looking at two traits simultaneously. Let's say that I have a parent who is purebred, yellow-eyed with wings. Yellow eyes and wings. And let's say they mate with somebody who is pure breeding recessive, white eyes and no wings. What the law of independent assortment says is just because you get a dominant version of A from one parent doesn't mean you're going to get the dominant I from a parent, that they're going to act completely independently of each other. Do you have any idea what to do with this? How to put this into a Punnett square? So I'm going to pull a real math teacher thing on you guys and start right with the hard stuff. I'll show you the trick. The hard stuff also happens to be the way biology teachers like to teach it because it makes y'all look smart when you can do it. But I want you to do it the easier way. Okay. So I'm going to show you this one. So what we're going to make is something called a dihybrid cross. Dihybrid crosses, or trihybrid or quadrihybrid, don't ever do those, are going to essentially determine all the possible gamete combinations and then see what the offspring could be. Okay, it's just like every other Punnett square. We're going to figure out what the gametes are, put them on the top, put them on the side, figure out the ratios of the offspring. But we can't make it easy. We wouldn't want that now, would we? All right, if you have a parent who is big A, big A, big I, they're going to get one copy of the A allele and one copy of the I allele. What are their gametes going to contain? Guaranteed. What version of the A allele will they definitely put into their gametes? J. What version of the A allele will this parent here have in all of their gametes? 100% of their gametes will contain big A. How about for the I allele or for the I gene? They'll all contain big I. So we know that this parent 100% has to have the big A and the big I in their gametes. So gametes are haploid. They only have one copy of each gene. That's what I've got here, a copy of the A gene and a copy of the I gene. How about for the other parent? We know that their gametes are going to be little a, little I. So we know that their offspring have to be big A, little a, big I, little I. Trust me, if you're going, what? We're going to do a lot of practice with this next week, so don't be in panic mode. I mean, you can be, but it's not going to do you much good because we'll get more practice. This is where you're going to hate me though. What if the parents were big A, big I, little I? What if they were hybrids? How can we figure out the genotypic ratios and the phenotypic ratios of offspring if what I've got is big A, big I, little I, mixed with another of the same? Any ideas on where to start? Say it again. So would it be possible for these two individuals to make an offspring that has the recessive traits? Well, we know that the law of segregation means that those traits are still potentially there. So they can't just use the dominant. We can't disregard something that's there. What do you think? Big A, big I, and then you have big A, little I. And so what do each of those combinations that you just said, what do each of them represent? What's that? Yeah, we're just going to do the same thing. If we were only looking at one trait, we'd say, okay, what are the gametes going to have? Ignore this example for just a minute. I'll erase this. I would not put this on your paper, right? But if I know somebody's, for example, big B, little b, I know that half of their gametes are going to contain the big B allele and half are going to contain the little, which is why I can set up my Punnett square, because we know that half of the offspring will get this big B from them and half will get this little b from them. And if the other parent is big B, little b, when I kind of distribute those out and show the possible fertilization combinations, it's the same thing. So now I just got to do it on a little bit larger scale and include both types or both genes. Okay, so if you are somebody who is big A, big I, little I, you're going to give half of your gametes the big A allele. Okay, you're going to give half of them the little a allele. You're going to give half of your gametes the big I allele, and half the little I. So that means I'm going to have 4 possible combinations. Okay, either big A, big I, big A, little I, little A, big I. Oh goodness, just look at the screen. I can't say all this, my brain hurts. Okay. These are the gametes that could arise from this parent. Do you all see that so far? So what could arise from the other parent? What do you think? What could the other parent give in their gametes? Oh, exactly the same thing. And so now all I have to do is a Punnett square. A really big Punnett square. So we're looking now at 2 different genes and trying to figure out what combinations of those genes could the offspring get. So I'm going to give you a couple of minutes. I'll do the top row. Then I want you to figure out the rest of it and figure out all of the phenotypic ratios. In other words, what phenotypes could there be? Okay. So for the top row, you would end up with AA, big A, big A, and big I, big I. Then you'd end up with big A, big A, big I, little I, because the second column has a little I there. Okay. Do you need help continuing on, or do you think you can just take a second and do this? All right, so I'll give you a couple of minutes here. So when we're looking at the law of independent assortment, the reason it's important to remember this is that we can essentially treat both traits as if they're completely unrelated to each other, right? Eye color or the whites of the eyes don't affect whether or not you have wings based on this law. And so I can basically just treat them separately. A dihybrid cross is nothing more than Punnett squares embedded in Punnett squares. So we figured out all the possible gametes. Check this out. These are both big A. These are both big A. If I just figure out the A's, what I'm going to have is that this first quadrant is all homozygous dominant. The next quadrant is going to all be heterozygous. The next quadrant is going to cross a big A with a little a. These are all going to be heterozygous. And finally, we're going to have all homozygous recessive. So far, so good? And so for those, all of them are going to have yellow eyes if they have the big A. Wow, that's far too large. Let me try that again. All of these are going to be the yellow phenotype, and the only ones that are going to be homozygous recessive will have white. I said that backwards, but you get the gist of it. Then within that Punnett square, I did 4 little Punnett squares. Big I, little I, big I, little I, big I, little I, big I, little I. True? So this would be, I don't even know what I'm doing now. There we go. Big I, big I. Okay, so the only ones that are going to not have wings are this one. So I'll use the crossed off to represent that they don't have wings. Don't do what I'm doing right now and be lazy with it. You still want to make sure that you can tell what your letters are. So all I did was just a Punnett square within a Punnett square. And if I wanted to do this with the tri-hybrid cross, guess what I would do? I would just have Punnett squares within Punnett squares within Punnett squares. Maybe that'll be a challenge at some point, but that is not something I would expect you to do. So when I need to count these out, yellow and winged, I'm looking for anything that is highlighted yellow that I don't have crossed out. There's going to be 9 individuals. So 9 out of the 16, I've got a 9 in 16 chance of getting the phenotype of yellow and winged if these were the parents, right? And then going on to yellow with no wings, there are 3 of those boxes that are highlighted but crossed out. So that's 3 out of the 16. 3 out of the 16 are going to be white and winged, and 1 out of the 16 are going to be both recessive traits. Yay, you learned the hard way. And guess what? You are going to have to do, you are going to have to do this several times in practice, but we'll be able to practice with it. Anyone want to know the easier way to do it? If these traits are truly independent and they assort independently, then the ratios will be just a product of simple probability. I say product quite literally. So the parents are big A, or sorry, big A, little a, big A, little a, big I, little I, big I, little I. If I were to do these Punnett squares out separately and figure out the phenotypes, write everything in here. What ratio would be expected to have the dominant phenotype? In this Punnett square, how many have the dominant phenotype? So yellow eyes, 3 out of 4. And how many have white? And then for winged, why did I write A there? Somebody's got to yell at me for doing stupid things. Just kidding. I'm sensitive, I'd cry. Right, and if this is going to be exactly the same, what ratio is going to have wings? And no wings would be 1 out of 4. So if I want to find out what percentage of my offspring would be expected to be yellow and winged, all I need to do is multiply those together. Yellow and winged is going to be 3/4 by 3/4, or 9/16. So what percentage or what ratio would be expected to be yellow with no wings? Okay, so we're going to now do 3/4 by only a 1 in 4 chance of getting one that doesn't have wings, which means that this combination of both of these two things is going to be 3/16. White winged, same thing. We're going to have 3/16. And then both recessive, I've got a 1/4 chance of being white, a 1/4 chance of not having wings. Multiply those together, and that gives me that 9-3 to 3-1 ratio that we just saw. Okay. We are going to practice this a lot next week. But you did the Punnett squares. You calculated the ratios that are expected in your gametes. Now it's time to find out what you actually get. Because what better time than Valentine's Day weekend to make some babies? Okay. All right. So again, this is all just the probability. This is telling you the likelihoods of what your kids are going to end up looking like. But now let's really do it. If you go in Schoology, stop this share. Yes. Is it like very recent or? Yep, yeah, got it. Yeah, for sure. Sorry, I'm trying to get a little bit too many things going at once. All right, so we're going into Schoology. And let me just talk really quickly about what you should have had from yesterday, just to make sure that you are good with that. If you did not get a chance to do yesterday's activity, you can still contribute to it and do that whole route. So this is where it gets ugly. Hopefully it stays sharing. We'll find out. All right. So if we take a look at yesterday, you guys went through the process of retrieving the gametes. So you guys made gametes already. That was meiosis. That whole purpose was to make the cells that would then become half of the genetics for a baby, for an offspring, right? So if we're taking a look here, I just want to point something out. You should have the parent genotype, so that's you, whatever your genotype was, and the finished gametes. So if you uploaded just your genotype, but the finished gametes are not in Google Drive yet, please make sure to upload that before you do anything else. That should be like the first thing you do. So for example, if I'm taking a look, I'm not going to take a look at anything apparently. I'm just going to, there we go. Did you do that, Juliana? Did you have both slides on yours with the gametes as well?
Yep.
Good. So I'm going to open this one up. So what you should see is something that looks like a spinning wheel of death forever. Oh, just kidding. Okay, I'm done. So here is the parent genotype. This is good. Juliana, I didn't know if you were going to become a Julian, if you were a dude. This is the parent. And that parent went through meiosis and made 4 different gametes. Now in dragons, they only make 4 gametes. We make billions, but dragons only made 4. So we retrieved them. We have them on a petri dish. Our goal today is to actually make the offspring, make the babies. So what your homework assignment over the weekend is to do is, if you go into the Schoology folder called Heredity, I think I put it in here. What's that do? Making babies activity, and go to day 2. We're back at the clinic. Yay, it worked. And so we're going to be retrieving our embryos. We're not retrieving our embryos. Selecting the gametes to make the embryos. And the only part that I think is a little bit confusing that I really want to clarify on is this right here. We have to have a way to randomly select a gamete. We don't want to just go, oh, that one has like wings and I want to make sure that my kid gets the allele for wings. Au contraire. You want to make sure that you are selecting randomly, because that's an important part of sex. The whole point of having sex is genetic variability. So we maximize out the randomness. So you're going to start by numbering them. I give you a system here to do it. If it doesn't work for you, I don't care. Pick a way to number your gametes. Pick some way. And then number them 1 to 4 before you select using the die, which it says, click on this, it'll bring you to that. You'll see a thing, it'll do a thing. Oh, no, oh, wow, we got a 7, that's very confusing. But the 4-sided die rolls a 1. So that's what we want to see, is that 4-sided die picking 1 through 4. When all is said and done, what I want from you on Tuesday is as follows. The gamete of one parent, the gamete of the other parent, and then a picture of your baby. If you want to make a whole family portrait, you can. That'd be super cute. But at least show me a picture of the baby. This can be done on paper. This can be done digitally, but I do want to make sure that you have the gametes and show all of the alleles that are on that gamete, okay? So once you have picked out which one it is, randomly, start figuring out your genotype, your phenotype, and making the baby. Okay, am I too excited? What can the baby look like? I don't care, so long as it follows the guidelines. So if it says that it needs to have such and such color skin, make it happen. If you suck at drawing, it's okay, I'm not judging you based on that. As long as somewhere in there I can be like, oh yeah, those are wings, baby should have had wings, we're Gucci, okay? Also, if your partner doesn't know that they're your partner and you're taking their gametes, We'll let them know on Tuesday, but I feel like that'll be funny to be like, haha, I made a baby with you and I know you didn't know it, but we did. So that was super fun. Okay. So you don't necessarily need to have a partner that you're working with. You can just have someone that you're taking their sperm or egg from. Okay. Do you have any questions about that?